Genetics has two categories, qualitative genetics and quantitative genetics. Qualitative genetics are characteristics that can be one thing or another like red or blue. There's no gradation between them. Mendel studied these traits. Quantitative genetics vary continuously with a large range of gradation like weight and height. Phenotypes are usually controlled by several genes, and normal distribution is a bell curve.
A quantitative trait is caused by polygenic inheritance. Polygenic inheritance is when one characteristic is controlled by multiple genes. This can be divided into three factors: additive genes, epistasis, and the dominance of the alleles. Polygenic inheritance can result in many different genotypes, but can all have different phenotypes or they can share one phenotype. The environment can also have an effect on quantitative traits.
There are three types of quantitative characteristics: continuous characteristics, which can assume any value between two extremes; meristic characteristics, which are non-continuous, determined by multiple genetic and environmental factors and measured as whole numbers; and threshold characteristics, which are either present or absent, where an underlying risk determines expression.
Quantitative genetics is based on statistics. Continuous traits fit into a normal distribution curve, which shows the mean of the data, or the center of distribution, variance, or how far the data is from the mean, and the standard deviation, which is the square root of the variance. On a distribution curve, 95% of the data should fit under the mean plus or minus 2 standard deviations. Covariance is the measure of how much two random variables change together. Correlation is when a change in one variable is associated with a change in another variable. Covariance is used to quantify the correlation. The correlation coefficient measures the strength of two variables' correlation using their covariance and their standard deviations.
Phenotypic variance is the diversity of statistical phenotypes in a population. The equation is total phenotypic variance equals genetic variation plus environmental contribution plus genetic and environmental interaction, where gene expression depends on the environment. Genetic variance is further divided into three parts: additive variance, or additive effects of genes on a phenotype; dominance variance, which masks recessive alleles at a locus; and interaction variance, which is based on epistasis and relations between genes.
Heritability is the proportion of the total phenotypic variation that is due to genetic differences. There are two types of heritability: narrow sense and broad sense. Broad sense heritability is the ratio of total genetic variance to total phenotypic variance. Narrow sense heritability is the ratio of additive genetic variance to the total phenotypic variance. Regarding heritability, there is no universal heritability for a characteristic. It only indicates differences among populations, not individuals, and is specific to the population and environment you are analyzing.
Locating genes affecting quantitative characteristics. Quantitative trait loci, or QTLs, are chromosomal regions containing genes that control polygenic characteristics. These are not genes. Genomic markers like SNPs can help find QTLs in the genes they affect. Genome-wide association studies look for associations between traits and sites of alleles in a biological population. Using this is more effective than the traditional method of crossing varieties that differ in a quantitative trait.
Both natural and artificial selection can have an effect on variable traits. Response to selection is the extent to which a characteristic is subjected to selection. This is influenced by narrow sense heritability and how much selection there is. Response to selection may eventually level off, and the variable characteristics may no longer respond to selection.